Coming to our today's topic, lipid chemistry. So what are lipids? Well, lipids are defined as the heterogeneous group of compounds which are relatively insoluble in water, means polar solvents, and they are soluble in the non-polar solvents like benzene, ether, chloroform and acetone. So simply you can say that lipids are the compounds which are insoluble in water and soluble in the non-polar solvents, and they have a variety of compounds. That's why we call them a heterogeneous group of compounds. And these lipids have clinical application in our body. Whenever there is excess accumulation of fat in the body, it leads to obesity. And apart from that, whenever there is increased cholesterol levels in the blood and also lipoproteins in the blood, it leads to atherosclerosis and cardiovascular diseases. That is one more clinical significance of lipids in our body. And when you see in case of diabetes mellitus, and there is a dearranged fatty acid metabolism, there is increased fatty acid oxidation, and that leads to a condition called as ketosis. So these are the important things what happen in case of excess amount of fatty acids and dearranged fatty acid metabolism. Coming to classification of lipids, lipids are classified into three groups, and this is proposed by Blues. So according to Blues classification, lipids are classified into three groups: simple lipids, compound or complex lipids, and derived lipids. And this classification is based on their composition. Simple lipids, the name itself indicates that there is simple composition and they are esters of fatty acids with alcohol. So their composition is they have alcohol and fatty acid. Right? So based on the type of alcohol, again simple lipids are subclassified into two types. Those are fats and oils and waxes. Coming to fats and oils, these fats and oils they have the composition of alcohol plus fatty acid, and the alcohol in case of fats and oils is glycerol. And when you see in case of waxes, they also have alcohol plus fatty acid, but the alcohol in case of waxes is a higher molecular weight alcohol. So that is the basic difference between these two simple lipids, fats and oils and waxes. Both are simple lipids, but waxes have a alcohol different from that of fats and oils. So in case of waxes, we have a high molecular weight monohydric alcohol, and the example is, we can say, cetyl alcohol in the form of cetyl palmitate, and myricil alcohol, and that is in the form of myricil palmitates in case of beeswax. And the simple fats and lipids, and the example is triacyl glycerol. And the difference between fats and oils is, fats are solids at room temperature and oils are liquids at room temperature. So there is a physical difference between fats and oils. Both are a type of simple lipids. So this is about simple lipids and their composition. And now coming to, so as I told you, the simple lipid, one of the simple lipids, fats and oils, they have this triacyl glycerol. So a glycerol molecule esterified to the three fatty acids, and three carbons have three fatty acids, C1, C2 and C3. Right. Now coming to complex lipids, so there is some complexity in the composition of these lipids, and that complexity is, in addition to the alcohol plus fatty acid, they have some non-lipid part. So that's why these lipids are called as compound or complex lipids. So based on the type of that non-lipid part, we have subclassified these compound lipids into phospholipids, glycolipids, sulfolipids and lipoproteins. So these are the different types of compound lipids. Among them, phospholipids are very important. So what are phospholipids? Phospholipids are a type of compound lipids, and coming to their composition, they have alcohol plus fatty acid and a non-lipid part. So in case of phospholipids, the non-lipid part is phosphate group. So that's why they are called as phospholipids. And based on the type of alcohol present in the phospholipids, we have subclassified phospholipids into two types, and those are glycerophospholipids and sphingospholipids. If the alcohol is glycerol, we call it as glycerophospholipid, and if the alcohol is sphingosine, we call such type of phospholipids as sphingospholipids. So that is the subclassification of phospholipids based on their composition. And coming to examples for glycerophospholipids, the examples are lecithin, cephalin, ethanol, lecithin and cephalin. And coming to spingophospholipids, we have Spingo Myelin. So these are the examples for the glycerophospholipids and spingophospholipids of subclassification of phospholipids. And when you see the structure, in case of spingophospholipids, the backbone is alcohol, sphingosine, and in case of glycerophospholipids, the backbone is glycerol. And coming to one of the subclassification of phospholipids, glycerophospholipids. So glycerophospholipids are a type of compound lipids having glycerol as the alcohol and fatty acid plus phosphate group, right? So there are many glycerophospholipids that occur in our body, that are present in our body, and one of them is phosphatidic acid. The simplest phospholipid in our body is phosphatidic acid. So though it is in very less concentration in tissues, but it acts as intermediate in the synthesis of triacylglycerols and other phospholipids. So the simplest phospholipid is phosphatidic acid. When you see the structure of phosphatidic acid, it has a glycerol backbone, and there are two fatty acids esterified at C1 and C2, and the third carbon is esterified with a phosphor group or phosphoric acid group, right? So that's why this is called as simple phospholipid, phosphatidic acid. Now coming to another phospholipid, phosphatidyl ethanol amine. So when you see the phosphatidic acid, when you compare phosphatidic acid and phosphatidyl ethanol amine, the basic difference between these two phospholipids is, in case of, so to the phosphate group, in case of phosphatidic acid, if you add a ethanol amine group, then it becomes a phosphatidyl ethanol amine. So simply you are adding ethanol amine to the phosphate group of the phosphatidic acid. So that results in the formation of phosphatidyl ethanol amine. So this phosphatidyl ethanol amine is present in case of brain and nerve tissue. So those are the sites where it is present. And coming to phosphatidyl choline, another phospholipid, glycerophospholipid. So this glycerophospholipid, phosphatidyl choline, has a type of compound, nitrogenous base attached to this phosphate group. That nitrogenous base attached to the phosphate group is choline. So that's why it is called as phosphatidyl choline. So simply to the phosphatidic acid you are adding choline, that becomes a phosphatidyl choline, and this phosphatidyl choline is the most important phospholipid in our body. This is present in liver, and apart from that this is present in brain and nerve tissue. So these are the important sites where it is present, phosphatidyl choline. And coming to phosphatidyl serine, and here instead of choline you are adding serine to the phosphatidic acid, so then it forms the phosphatidyl serine. So that is the basic difference between phosphatidyl choline and phosphatidyl serine. Just you are adding serine, a hydroxy group containing amino acid, to that phosphate group, so that it results in the formation of another phospholipid, glycerophospholipid, phosphatidyl serine. And this is also present in the brain and nervous tissue. And there is few less amount of this phosphatidyl serine is also seen in blood. And coming to another phospholipid, glycerophospholipid, phosphatidyl inositol. So phosphatidic acid plus inositol forms phosphatidyl inositol, and this is about the phosphatidyl inositol. Now coming to another glycerophospholipid, Plasmalochen. So till now, from phosphatidic acid to phosphatidyl inositol, we have seen that the fatty acids are esterified to the C1 and C2 carbons of glycerol, and there they have ester linkage. But when you see in case of plasmalogen, the C1 of glycerol is esterified to a unsaturated fatty alcohol, so that's why we call it as plasmalogen. So that is the difference between remaining glycerophospholipids and this plasmalogen. So this plasmalogen has an unsaturated fatty acid attached to the glycerol by ether linkage instead of ester linkage, right? So this is the major when you see the concentration of plasmalogen when you compare with that of other phospholipids, so it constitutes about the 10% of all the phospholipids in the brain. So that is the significance of plasmalogen and its concentration. So another phospholipid, glycerophospholipid called as cardiolipin or phosphatidyl glycerol. So this is formed by combination of two phosphatidic acids, and those two phosphatidic acids are joined by a glycerol molecule. So that's why it is called as phosphatidyl glycerol or cardiolipin, having two phosphatidic acids linked by an additional glycerol molecule. So this is present in mitochondria, and this is the only phospholipid which has an antigenic property. So that is the significance of phosphatidyl glycerol. And so till now we have seen about the glycerophospholipids, a type of phospholipids, and the other subclassification in case of phospholipids is spingophospholipids. So these spingophospholipids, they have alcohol sphingosine, and in addition to sphingosine they have fatty acid and they have fatty acids. So sphingosine is the alcohol, and to that we have a fatty acid attached to that fatty acid. So that's why we call it as spingophospholipids, and the example for spingophospholipids is Spingo Myelin. So in case of Spingo Myelin, we have a sphingosine alcohol, to that fatty acid is attached, and there is a nitrogenous base choline. So this Spingo Myelin is mainly present in the myelin sheath, and that's why it is named as Spingo Myelin. So myelin is derived from its location, that is, it is present in the myelin sheath. So when you see this structure of spingomyelin, we have sphingosine plus fatty acid. So sphingosine plus fatty acid together we call it as ceramide. So ceramide is the structure present in the spingomyelin. It's nothing but sphingosine plus fatty acid. And that's about the one of the major class of phospholipids, compound lipids, that is phospholipids. Now coming to another class of phospholipids, glycolipids, and they are also called as glycospingolipids. So here, as I told you, so these glycolipids have another part, non-lipid part, and that non-lipid part is carbohydrate. So compound lipids, they have alcohol plus fatty acid plus a non-lipid part, and non-lipid part in case of glycolipids is carbohydrate. So their composition is sphingosine plus fatty acid plus carbohydrate. So based on the type of carbohydrate, we have subclassified these glycolipids into three types, and they are cerebrocytes, globocytes and gangliosides. So coming to cerebrocytes, so these are the simplest form of glycolipids, and when you see the structure of these cerebrocytes, they have a ceramide structure plus one hexosugar. It may be glucose, galactose or mannose, whatever it may be. So they have ceramide, means sphingosine plus fatty acid plus, and the carbohydrate is a single monosaccharide unit. So those are called as cerebrocyte. For example, you can take glucose cerebrocyte, galactosyl cerebrocyte. So based on the type of monosaccharide, we have different types of cerebrocyte. So these are the simplest forms of glycolipids. And coming to globocytes, so they are some more complicated types of glycolipids. These globocytes, they have cerebrocyte plus two monosaccharides, two or more monosaccharides. It is called as globocytes. So basic composition in case of globocytes is they have ceramide, means sphingosine plus fatty acid plus two or more monosaccharides. They may have glucose, galactose, they may have glucose, mannose, galactose, like that. They have two or more than two monosaccharide units. So such type of glycolipids, we call them as globocytes. And coming to the more complex type of glycolipids, gangliosides. So these gangliosides, they have basic structure again ceramide, means they have sphingosine plus fatty acid. In addition to this, they have two or more monosaccharides, simple monosaccharides, and in addition to these monosaccharides, they have a sialic acid called as N-acetyl neuraminic acid. So glycolipids which have sialic acid or N-acetylenonic acid in their structure, in their composition, we call such type of glycolipids as gangliosides. So these gangliosides are the most complicated types of complex type of glycolipids. So this is about the glycolipids, another type of compound lipids. Now coming to another type of compound lipids, again you see lipoproteins. Here the non-lipid part is proteins, and they have lipid part plus non-lipid part, and non-lipid part, when it is a protein, we call them as lipoproteins. So examples for lipoproteins are chylomicrons, VLDL, LDL, HDL. So all those are the lipoproteins. And apart from that, we have other types of compound lipids like sulfolipids, aminolipids, lipopolysaccharides. So all these are compound lipids. So this is about the classification of compound lipids. So we have classified compound lipids into phospholipids, glycolipids, lipoproteins, and other complex lipids. And the third type of lipids in classification is derived lipids. So derived lipids are obtained by hydrolysis of group 1 and group 2 lipids. Means, so when you subject simple lipids and compound lipids to hydrolysis, so whatever the products we obtain, we call those lipids as derived lipids. So examples for derived lipids are glycerol, alcohols, fatty acids, monoacylglycerol, diacylglycerol, and fat soluble vitamins. So all those come under these derived lipids. So basically, when you are subjecting, you are taking simple lipids and compound lipids and you are subjecting them to hydrolysis, and the products whatever you get after hydrolysis, we call them as derived lipids. So this is the classification of lipids into three types: simple lipids, compound lipids, and derived lipids. So when you see, we discussed that simple lipids are the esters of fatty acids with alcohol. So what are fatty acids? So fatty acids are the carboxylic acids with the hydrocarbon side chain. So they have a carboxylic group, to that hydrocarbon side chain is linked. So this type of organic acids which have carboxylic acids with hydrocarbon side chain, we call them as fatty acids. So most of the fatty acids, they have a carbon length of 14 carbons to 20 carbons, and the most predominant type of fatty acids in our body are palmitic acid and stearic acid. And these fatty acids can be classified into saturated and unsaturated fatty acids. So that depends upon the double bonds. So whenever a fatty acid has double bonds in its structure, we call them such type of fatty acids as unsaturated fatty acids, and fatty acids without the double bonds, we call them as saturated fatty acids. So we can classify fatty acids into saturated and unsaturated fatty acids based on the presence of double bonds. So with the double bonds, we call the fatty acids we call them as unsaturated, and without double bonds, we call them as saturated fatty acids. Right. So some of these are the some important saturated fatty acids: butyric acid, caproic acid, laric acid, palmitic acid. When you see their structure, so there is no double bond in them, so that's why we call them as saturated fatty acids. And coming to unsaturated fatty acids, so we have examples like oleic acid and linoleic acid. So when you see the structure of oleic acid, it has 18 carbons, and 18:1 indicates that this oleic acid has one double bond. So that's why it is a unsaturated fatty acids. So when unsaturated fatty acids have one double bond, we call them as monounsaturated fatty acids. If they have more than one double bond, we call them as polyunsaturated fatty acids. So we can see the other example, linoleic acid having 18 carbons, but it has two double bonds. 18:2 means it has two double bonds. Right. So at the positions between 9th and 12th carbons, we have two double bonds. So these are the two examples for unsaturated fatty acids having the double bonds in their structure. Now coming to the most important aspect of fatty acids is essential fatty acids. So there are some fatty acids which cannot be synthesized in our body, and they should be supplied in the diet. Such type of fatty acids are called as essential fatty acids. So those which cannot be synthesized in our body and should be supplied through the diet are called as essential fatty acids. So when you see examples, those are the linoleic acid and linolenic acid. So these two are called as essential fatty acids. And when you see their structure, they have more than one double bond, and these are called as polyunsaturated fatty acids. So polyunsaturated fatty acids like linoleic acid, linolenic acid are called as essential fatty acids. So what is the importance of these essential fatty acids? So these essential fatty acids, they have several important roles. So these essential fatty acids, they act as anti-atherogenic factors, means they prevent the atherosclerosis, they prevent the accumulation of lipids in the arteries, so they act as anti-atherogenic agents. So that is one most important function of essential fatty acids. And coming to another role, they have some important role in vision. That is, there is one fatty acid synthesized from essential fatty acids like docosahexaenoic acid. So that is synthesized from the linolenic acid, whatever you are taking the diet, linoleic acid. So that is converted into docosahexaenoic acid, and that docosahexaenoic acid is seen its concentration is more in case of photoreceptor cells. So retinal photoreceptor cells, they have high concentration of this docosahexaenoic acid. In short, we call it as DHA. So that indicates the role of fatty acids in the vision. So that is the one more important biochemical role of essential fatty acids, role in vision. So coming to clinical significance of essential fatty acids. So whenever there is deficiency of essential fatty acids, the concentration, it leads to a condition called as retinitis pigmentosa. So in case of retinitis pigmentosa, there is decreased levels of this docosahexaenoic acid seen in those cases. So that is the clinical significance of this DHA. And apart from that, see, this essential fatty acid, they have structural roles. So tissues, they have these essential fatty acids. Apart from that, these essential fatty acids are also required in the synthesis of prostaglandins. So we have discussed that essential fatty acids are linoleic acid and linolenic acid. So that linolenic acid is involved in the synthesis of another type of fatty acid called as arachidonic acids. So that arachidonic acid is synthesized from these essential fatty acids, and that arachidonic acid acts as a precursor for the synthesis of a type of compounds called as prostaglandins and leukotrienes. So that is one more role of essential fatty acids. And apart from that, these essential fatty acids are also involved in the maintain the serum cholesterol levels. So they decrease the serum cholesterol level. And the other role is the concentration of the essential fatty acids is more in case of gonads. So that indicates the role in the fertilization. And coming to another role, these essential fatty acids are present in lipids. So the membrane fluidity is because of phospholipids with essential fatty acids. So that is the significance of essential fatty acids in our body. So whenever there is deficiency of the essential fatty acid, it leads to some conditions like it leads to scaly dermatitis, there are skin manifestations, and it leads to poor wound healing, and there is hair loss seen in case of deficiency of essential fatty acids. And there are few tests to identify whether the given fat is pure or not, whether there is any adulteration or not. So to identify that, we will do some tests. So those are iodine number. So what is iodine number? So iodine number can be defined as, so number of grams of iodine required or absorbed by 100 grams of fat is called as iodine number. So when you take 100 grams of fat, so that absorbs a number of grams of iodine. So that is defined as the iodine number. So to be simple, so number of grams of iodine absorbed by 100 grams of fat is called as iodine number. And what is the significance of this iodine number? So this iodine number indicates the degree of unsaturation. So it indicates whether the given fat is unsaturated or not, and what is the degree of unsaturation in that given fat. And when you see examples, so when you do this test in case taking butter, it gives a number 28. So butter iodine number is 28, and in case of sunflower oil, so the iodine number is 130. So whenever butter gives 28 iodine number, so we can say that it has the required unsaturation, and when you see sunflower oil, it should give 130 iodine number, so then it indicates the normal unsaturation of sunflower oil. So whenever there is alteration in this range iodine number, it indicates some adulteration of those fats and oils. And another test for the identification of adulteration is saponification number. So what is saponification? Hydrolysis of fat by alkali is called as saponification. And coming to saponification number, so it can be defined as milligrams of KOH required to saponify 1 gram of fat is called as saponification number. So what is the significance of this saponification number? So this saponification number indicates the molecular weight of that fat. So that molecular weight of fat in turn depends upon the type of fatty acids. If the short chain fatty acids are more in concentration, so the iodine number will be altered. So the examples for saponification number are butter, it has a number saponification number of 210 to 230, and in case of coconut oil, it has a saponification number of 250 to 262. So whenever this saponification number increases, that indicates that there are more amount of short chain fatty acids. So these are the two tests used for the identification of adulteration in a given fat: iodine number and saponification number. And as per the definition, lipids are insoluble in water and they are soluble in organic solvents. But there are some few exceptions, like when you see phospholipids, bile salts, fatty acids, they have some hydrophilic groups, so that's why they are soluble in water. So such type of fats which have both hydrophobic groups and hydrophilic groups, we call them as amphipathic lipids. So they have both groups, hydrophobic and hydrophilic groups. And so this amphipathic nature of these lipids is utilized in some important formation of some important compounds like micelles and in the formation of liposomes. So when you see micelles, what are micelles? So these micelles are formed when you mix these amphipathic lipids in the aqueous solution. So that arranges those amphipathic lipids in such a fashion that so the periphery has the groups. So those groups which interact with the water, those are the hydrophilic groups, and the hydrophobic groups are arranged towards the interior of that micelles. So these are called as the micelles. So micelles are formed when you mix these amphipathic lipids in the aqueous solution. And coming to liposomes, so liposomes are also because of the amphipathic nature of lipids. So these liposomes are formed so when you mix these amphipathic lipids in the aqueous solution by sending sound waves. So you add the amphipathic lipids to the aqueous solution, and when you send the sound waves, it leads to arrangement or formation of liposomes. So the specificity of liposomes, the significance of liposomes is, so these liposomes have in their core aqueous solution, and that liposomes, they are arranged in the circles. So there are concentric circles of liposomes which within them they have the aqueous solution. So these liposomes and micelles are formed because of the amphipathic nature of the lipids. So these liposomes, they have some clinical significance. So there are some drugs. So those drugs are given by, they are placed in these liposomes, and they reach and they have more affinity, or they can have high significant mechanism of action. So when you compare with this the other ways in which you introduce these drugs. So when you add drugs to these liposomes, they are carried to the target tissues more efficiently than the other routes of administration of drugs. So that is a clinical significance of liposomes. So we have discussed about in lipid chemistry, we discussed about the classification of lipids, and what are fatty acids, what are essential fatty acids, their clinical significance and their functions, and what are the tests used for the identification of adulterate fats like saponification number, iodine number, and we have seen some important properties of lipids like amphipathic nature of lipids and their clinical significance. So this is about lipid chemistry. Thank you.